Okay, let's really get into it today. Unpack this incredible organ, the liver. I mean, forget just thinking of it as another part of the body. This thing is, well, it's the undisputed champion, isn't it? The body's central chemical factory.
It really is. Juggling hundreds of vital tasks all at once. It's like the ultimate multitasker.
Yeah, handling everything from, scrubbing your blood clean to fueling your metabolism, storing essential supplies. It's just constantly working.
It's true. The sheer scale and complexity are just, well, astonishing. And understanding it isn't just knowing what it does, it's really grasping the intricate how and why.
Exactly. And to get to that level, that kind of mastery that, the elite professionals have, we're diving deep into some serious source material today. We're using Guyton and Hall, which is like a foundational text in physiology.
Absolutely. It's solid stuff. Not exactly light reading, but it's packed with the precise details you need to truly understand the liver.
Right. Guyton Hall gives us that bedrock, the structure, the blood flow, how the cells work, the clinical side too. So our mission, really, for you, our listener, is to take all this expert-level detail.
And make it clear, make it stick. We want to pull out the most important insights and connect the dots so the complexity feels, well, manageable and actually pretty fascinating.
So we'll start from the ground up, look at the liver's unique anatomy, how it's actually built. Then we'll explore its incredible vascular system, that massive blood flow, how it handles fluid, which, leads to things like ascites.
Then its amazing power to regenerate, almost mythical, really.
Definitely. Then we'll dive into its metabolic superpowers, how it handles carbs, fats, proteins, basically driving the body's whole energy economy.
We'll also cover its role storing things like vitamins and iron, which is crucial.
And finally, we'll use bilirubin, that pigment, to show how understanding these basic processes is actually a powerful diagnostic tool in medicine.
It's quite a journey into body chemistry and function. Shall we start with the foundations? The physiological anatomy?
Let's do it. So starting big picture, size and scale. What does Guyton Hall say?
Well, the first thing is just how big it is. It is, without question, the largest internal organ in your body.
Largest internal organ.
Yeah.
Okay, so it's physically substantial. What does that translate to in, weight?
In an average adult, it makes up about 2% of your total body weight,
2%.
which works out to roughly 1.5 kilograms or, around 3.3 pounds.
1.5 kilos. That's a serious amount of tissue dedicated to these functions. How is all that mass organized? The source mentions a basic functional unit.
Yes, and this is really key to understanding how it works at the micro level. The basic functional unit is called the liver lobule. You can sort of picture it as a tiny cylinder.
A tiny cylinder. How tiny are we talking?
Yeah.
Dimensions.
Guyton Hall describes it as cylindrical, several millimeters long, and maybe 0.8 to 2 millimeters in diameter. So individually, they're quite small.
But I imagine the power's in the numbers, right? Like lots of them together.
Exactly. The human liver contains a huge number, somewhere between 50,000 and 100,000 of these individual lobules.
Wow.
Think of it like a massive city built from tens of thousands of these tiny self-contained processing units all working together.
50,000 to 100,000 tiny factories packed into that 1.5 kilos. Okay, let's zoom in on one lobule. What's the internal layout?
So the structure is, well, quite elegant, really. It's built around a central vein. This vein runs right down the middle of the cylinder.
Okay, a central vein. And where does that lead?
It ultimately drains into the larger hepatic veins, which then carry the blood away from the liver and into the vena cava, the body's main vein heading back to the heart.
Got it. So the central vein is the exit point for blood. What radiates out from it?
Radiating outwards from that central vein are what Guyton and Hall call liver cellular plates. These are basically thin sheets of liver cells, the hepatocytes.
How thick?
Typically just 2 cells thick. They extend out from the central vein, kind of like spokes on a wheel or maybe walls in a honeycomb pattern.
Sheets of cells, just 2 thick, arranged radially. Interesting. What about the spaces between the cells within those plates?
That's where the bile drainage system starts. Between adjacent liver cells, right there within the plates, you find these tiny channels called bile canaliculi.
Canaliculi.
This is literally where the liver cells secrete bile, and it starts its journey out of the liver.
And where do these tiny bile channels drain?
They don't just open into space, they collect and empty into larger bile ducts, and these ducts are located in the fibra septa.
The septa. Those are the walls between lobules.
Exactly. Thin walls of connective tissue that separate one lobule from the next.
Okay, so bile's made in the plates, flows into tiny channels between the plate cells, then empties into bigger ducts between the lobules. Makes sense. Now, the blood flow, this is where it gets really unique, right?
Oh, absolutely. In those same fibrous septa where you find the bile ducts, you also find small portal venules.
Portal venules.
Yeah.
Venules meaning small veins. So this is blood coming in.
Precisely. This is blood arriving mainly from the venous outflow of your entire digestive system, the stomach, the intestines, via the main portal vein. It's carrying all those freshly absorbed nutrients and, well, anything else absorbed from the gut.
Right. So loaded blood arrives in these portal venules. Where does it go next within the lobule?
From these venules, the blood flows into these remarkable vessels called hepatic sinusoids. These lie between those liver cellular plates we talked about.
The sinusoids. I've heard these aren't like typical capillaries.
Not at all. They're described as flat, sort of branching vessels. The blood flows through them, winding its way between the plates of liver cells, heading inwards towards that central vein.
So the blood is flowing past the liver cells.
And the absolutely critical point Guyton and Hall emphasizes here is that the liver cells, the hepatocytes, are continuously exposed to this portal venous blood flowing through the sinusoids.
Constant exposure. That seems vital. The liver cells are right there, ready to grab and process whatever's coming in from the gut instantly. But doesn't the liver also get oxygenated blood from the hepatic artery?
Yes, it does, and this adds to the unique mix. You also have hepatic arterioles located in those same interlobular septa.
Okay.
These supply oxygenated arterial blood primarily to the septal tissues themselves, but importantly, Guyton and Hall notes that many of these small arterioles also empty directly into the hepatic sinusoids.
Directly into the sinusoids. So the sinusoids contain a mix of blood.
Exactly. The source mentions they often empty in about 1/3 of the way from the septa towards the central vein. So you get this unusual mixture. Nutrient-rich, potentially toxin-laden venous blood from the gut mixed with fresh, oxygen-rich arterial blood, all flowing together through the sinusoids, constantly bathing the liver cells.
Wow, okay, so we have the cell plates, the bile channels starting between cells, this unique mixed blood flow in the sinusoids between the plates. What about the lining of the sinusoids themselves? The source mentions special cells.
Yes, the lining is quite special. It's made up of typical endothelial cells forming the vessel wall, but scattered amongst them are these large specialized cells called Kupffer cells.
Kupffer cells. These are the liver's immune cells, right? The macrophages.
Precisely. Guyton and Hall identifies them as resident macrophages, a key part of the body's reticuloendothelial system. They are strategically positioned, lining the sinusoids.
And their main job.
Their primary mission is phagocytosis, basically engulfing and destroying bacteria, cellular debris, and other foreign particles that come flowing through in that portal blood. They're like the liver's bouncers, checking everything coming in from the gut.
A crucial defense line, given what comes from the intestines. The source also highlights something really unusual about the endothelial lining itself, the non-Kupffer cells. Something about pores.
This is another fascinating detail with huge functional consequences. The endothelial lining of the sinusoids is riddled with extremely large pores or openings. Guyton and Hall state some are almost 1 micrometer in diameter.
1 micrometer. How does that compare to, say, normal capillaries?
It's vastly larger. Capillaries elsewhere have much smaller pores, mainly letting fluid and small molecules pass through. These massive pores in the liver sinusoids are a completely different ballgame.
What's the significance of having such leaky walls in the sinusoids?
It leads directly to the formation of something called the spaces of Dissy. These are very narrow tissue spaces located just underneath the leaky endothelial lining, filling the gap between the endothelial cells and the actual liver cells, hepatocytes. They're also called perisinusoidal spaces.
So a gap between the blood vessel lining and the liver cell itself. And because the lining is so porous?
Substances in the blood plasma can move freely out of the sinusoid and into these spaces of Dissy. Guyton and Hall is very specific here. Even large portions of the plasma proteins diffuse freely into these spaces.
Even the big protein molecules. That's radically different from most capillaries where proteins mostly stay in the blood.
Exactly. It means the fluid in the spaces of Dissy is very similar to plasma, rich in proteins.
Wow. So the sinusoids are like leaky sieves, letting plasma fluid and even proteins bathe the outside of the liver cells in this space of Dissy. What stops this space just filling up and swelling?
That's where the lymphatic system plays a crucial role. Millions of these tiny spaces of Dissy connect directly with lymphatic vessels, which are located back out in those interlobular septa.
Oh, okay.
So the excess fluid, and crucially those proteins that have leaked out into the spaces of Dissy, are efficiently collected and removed by these lymphatic vessels. This high permeability, combined with very efficient lymphatic drainage, is fundamental to how the liver handles fluid.
Okay, so the anatomy paints a picture. Lobules with radiating plates of cells, unique mixed blood flowing through leaky sinusoids lined with filter cells, Kupffer cells, leading to protein-rich fluid bathing the liver cells in the spaces of DISI, which is then whisked away by lymphatics. It's a design optimized for intense, rapid interaction between the blood and the liver cells.
Absolutely. Every feature seems geared towards high-volume processing, filtration, and metabolic exchange.
That anatomical picture leads perfectly into the next section, the hepatic, vascular, and lymph systems. Let's talk about blood flow volume and resistance. Guyton Hall makes a striking point right away.
Yes, it emphasizes that the liver has incredibly high blood flow, but normally the resistance to that flow is very low.
High flow. Just how high are we talking? What are the numbers?
The total blood flow through the liver is about 1,350 milliliters per minute. That's quite a lot.
1,350 milliliters in a minute. How is that split between the portal vein and hepatic artery?
It's roughly 150 milliliters per minute coming from the portal vein, that's the blood from the gut, and an additional 300 milliliters per minute coming from the hepatic artery, providing the oxygenated supply.
Okay, 1,350 milliliter in total. How does that stack up against the rest of the body's circulation?
Guyton Hall puts it in context. This represents about 27% of the resting cardiac output.
Wow, more than a quarter.
Yes, over a quarter of all the blood your heart pumps each minute, while you're just sitting there, goes straight through the liver. Considering the liver is only about 2% of body weight, that's a disproportionately huge share. It really highlights its metabolic activity and central processing role.
Over a quarter of your blood flow. That's a stunning statistic.
Now, you mentioned low resistance. How is that reflected in the blood pressure across the liver?
The pressure drop is surprisingly small. The pressure in the portal vein entering the liver is typically around 9 millimeters of mercury, mmHg.
Okay.
And the pressure in the hepatic veins leaving the liver, heading towards the vena cava, is normally very close to 0 millimeter Hg.
So just a 9 millimeter Hg pressure difference is driving that massive 1,350 millimolar flow through all those sinusoids.
Exactly. That small gradient is enough to push that huge volume of blood through. It clearly indicates that, under normal conditions, the resistance to flow through the hepatic sinusoids must be very low.
But things can go wrong. What happens when that low resistance increases? Guyton and Hall immediately brings up cirrhosis. Right. Cirrhosis is the classic example where liver vascular resistance shoots up. It's a condition where the main functional liver cells, the parenchyma, are progressively destroyed.
And what takes their place?
They get replaced by fibrous scar tissue. And this isn't just passive scarring. Guyton and Hall explains that this fibrous tissue actually contracts around the blood vessels within the liver.
Like squeezing the pipes.
Precisely. It squeezes them, greatly impeding the flow of portal blood through the liver's vascular network.
So the plumbing gets clogged and constricted by scar tissue? What are the most common culprits causing this scarring, this cirrhosis, according to the source?
Guyton Hall lists several key causes. Chronic alcoholism is a major one. Another that's becoming increasingly common is excess fat buildup in the liver, which then triggers inflammation. This is known as non-alcoholic steatohepatitis, or NASH.
NASH. Yeah, we hear a lot about that. Does the source mention the related condition, NAFLD?
Yes, it mentions non-alcoholic fatty liver disease, NAFLD, which is a sort of precursor or less severe form involving just the fat accumulation without the major inflammation yet. Guyton and Hall specifically notes that NAFLD is extremely common in industrialized nations like the U.S. and is usually linked very strongly with obesity and type 2 diabetes.
So lifestyle factors like diet and weight are major drivers here. What other causes of cirrhosis are mentioned?
Cirrhosis can also result from exposure to certain toxins or poisons like carbon tetrachloride. Viral infections like chronic infectious hepatitis are another cause, and problems with the bile ducts like chronic obstruction or infection can also lead to cirrhosis over time.
So various insults can ultimately lead to this scarring and obstruction. That covers the gradual blockage. What about a sudden acute blockage of flow?
That can happen too. Occasionally, the main portal vein itself, or one of its major branches, can get suddenly blocked, for example, by a large blood clot.
And what's the immediate critical consequence of any kind of severely impeded flow, whether it's gradual from cirrhosis or sudden from a clot?
The major consequence is portal hypertension. That means the pressure builds up in the entire portal venous system before the liver. Guyton and Hall states this high pressure transmits backwards, raising the pressure inside the capillaries of the intestinal wall significantly, maybe 15 to 20 millimeter Hg above their normal pressure.
15 to 20 millimeter Hg higher.
Yeah.
That sounds like a lot for delicate capillaries in the gut. What's the severe immediate risk if a sudden total blockage isn't fixed quickly?
Guyton Hall paints a grim picture. If a sudden, complete blockage isn't relieved, the patient can actually die within just a few hours.
Why so fast?
Because that extreme pressure forces excessive amounts of fluid to leak out of the intestinal capillaries. This fluid floods into the walls of the intestines and even into the intestinal lumen itself. It's basically a massive fluid loss from the circulation into the gut.
Like the pipes bursting upstream due to the blockage downstream. A powerful example of why that low resistance is so vital. Okay, let's shift gears slightly, but still within the vascular system. The liver's role as a blood reservoir.
Yes, this is another interesting aspect. The liver is quite an expandable organ. And because it has such an extensive network of veins and sinusoids, it can actually store a surprisingly large amount of blood.
How much does it normally hold?
Under normal circumstances, the blood volume within the liver's hepatic veins and sinusoids is about 450 milliliters.
450 milliliters, that's nearly half a liter. How does that compare to the body's total blood volume?
It's close to 10% of your body's total blood volume just sitting within the liver at any given moment.
Wow, 10% already. Can it hold even more if needed?
Yes, it can expand significantly. Guyton and Hall notes that when pressure rises in the right atrium of the heart, which can happen, for example, in heart failure, this causes back pressure through the vena cava and into the hepatic veins.
Okay.
This back pressure makes the liver swell, and it can store an additional 0.5 to 1 liter of extra blood within its vessels.
So potentially holding over 1.5 liters of blood in total when under pressure, that's a huge buffering capacity.
It is. Guyton and Hall specifically mentions this occurs in cardiac failure with peripheral congestion. The liver essentially acts like a sponge, soaking up some of that excess blood volume to relieve pressure elsewhere in the circulation.
It's like a temporary holding tank for the cardiovascular system. So it can store blood when there's too much, and I assume it can release blood when the body needs more circulating volume.
Precisely. Guyton and Hall summarizes it perfectly. The liver is a large, expandable venous organ serving as a valuable blood reservoir in times of excess volume and capable of supplying extra blood in times of diminished volume. It helps buffer your overall blood volume levels.
A glucose buffer, a blood volume buffer. The liver really is all about maintaining balance. Now, related to that fluid handling and the leaky sinusoids we talked about, the liver also has incredibly high lymph flow, doesn't it?
Yes, this ties directly back to those unique sinusoids and the spaces of Dis. Guyton and Hall reiterates that the pores in the sinusoid lining are extremely permeable, much more so than capillaries elsewhere. This allows ready passage of not just fluid, but also, critically, proteins out of the blood and into those spaces of DISI.
And because proteins leak out so easily, what's the lymph draining from the liver like? Is it protein-rich?
Remarkably so. The source gives a figure. The protein concentration in liver lymph is about 6 grams per deciliter, GDL. That's only slightly less than the protein concentration in blood plasma itself. Very different from the low protein lymph found in most other tissues.
So valuable plasma proteins are constantly flowing out into the spaces around the liver cells. How much lymph does this high permeability generate?
It leads to the formation of large quantities of lymph. And here's a really striking fact from Guyton Hall. Under resting conditions, about half of all the lymph formed in your entire body actually originates in the liver.
Half. Half of all your body's lymph comes from the liver. That statistic really drives home the sheer scale of fluid processing happening there. Okay, let's connect this high permeability, the fluid flow, and potential high pressure to a major clinical issue, ascites.
Yes, ascites, that's the abnormal accumulation of large amounts of free fluid within the abdominal cavity. Guyton and Hall explains this is directly caused by high hepatic vascular pressures.
Which specific pressures are we talking about?
It can be high pressure backing up into the liver from the portal system, like in cirrhosis, or high pressure in the hepatic veins backing up from the vena cava, like in some types of heart failure.
Does it take a massive pressure increase to cause this fluid leakage into the abdomen?
No, and that's another important point. Guyton and Hall states that only a relatively small increase in hepatic venous pressure, just 3 to 7 millimeter Hg above normal, is enough to cause excessive amounts of fluid to transudate.
Where does the fluid go?
It goes into the lymph, as we discussed, but also, critically, it leaks directly through the outer surface of the liver capsule itself, dripping right into the abdominal cavity.
Leaking straight off the liver surface. Wow. What kind of fluid is it?
The source describes it as almost pure plasma. It contains 80% to 90% as much protein as normal plasma because of that high permeability we talked about.
So it's protein-rich fluid essentially sweating off the liver. Guyton and Huddle gives a more severe example.
Yes, they state that if vena cava pressure rises significantly, say to 10-15 millimeter Hg, hepatic lymph flow can increase up to 20 times normal. The amount of fluid sweating from the liver surface can become so great that large volumes of free fluid collect in the abdominal cavity that's severe ascites.
Sweating plasma, a vivid image.
Yeah.
And you mentioned ascites can also happen with portal blockage, like in cirrhosis. How does that work?
Exactly. Guyton and Hall explains that blockage of portal flow through the liver also causes high capillary pressures back in the entire portal system, meaning the capillaries in the walls of the intestines and stomach. This high pressure causes edema or swelling of the gut wall itself, but it also causes significant transudation of fluid through the serosa, the outer lining of the gut, directly into the abdominal cavity. So leakage from the gut surface also contributes heavily to ascites in cases of portal hypertension.
So ascites results from high pressure forcing protein-rich fluid out either directly from the liver surface due to back pressure from the heart side, or from both the liver surface and the gut surface due to back pressure from portal obstruction within the liver. It's all about fluid being squeezed out. Okay, we've covered structure, blood flow, lymph. Let's move to something truly amazing: the liver's ability to regenerate.
This really is one of the most remarkable features of the liver. Guyton Hall calls it a remarkable ability to restore itself after tissue loss.
When does this regeneration typically kick in?
It happens after significant loss of liver tissue. This could be from a surgical procedure called a partial hepatectomy, where a piece of the liver is removed, or it could be after acute liver injury caused by toxins or other short-term insults.
Is this regeneration always successful? Are there limitations?
Yes, and this is a critical caveat highlighted by Guyton and Hall. The regeneration process works well as long as the injury is uncomplicated by ongoing factors like viral infection or chronic inflammation.
Ah, okay.
So while its potential is incredible, things like chronic hepatitis or persistent inflammation can severely hinder or even prevent this regeneration.
That's a really important distinction. It's powerful but not invincible against chronic problems. How much liver can actually be removed and still regenerate effectively?
The source notes that surgeons can remove up to 70% of the liver.
70%.
Yes, and the remaining lobes will then enlarge dramatically, ultimately restoring the liver back to its original size and mass.
How fast does this happen? Is it a slow process?
No, Guyton and Hall describes it as remarkably rapid. They mention experiments in rats where the liver mass is fully restored in just 5 to 7 days.
Less than a week in rats.
Yeah.
That scale and speed is remarkable. What are the individual liver cells, the hepatocytes, actually doing during this rapid growth?
They undergo rapid cell division mitosis. Each hepatocyte might replicate just once or twice. But crucially, Guyton and Hall states they then stop dividing and revert back to their usual quiet, non-dividing state once the liver has regained its original size and volume.
They know when to stop. That's fascinating. How is this incredibly precise and rapid process controlled? What tells the cells to start dividing like crazy and then to hit the brakes?
Well, this is an area where, as Guyton and Hall acknowledges, the control mechanisms are not completely understood. However, they point to one key player that appears important: hepatocyte growth factor, or HGF.
HGF, okay. Where does it come from? The liver cells themselves?
Interestingly, no. The source says HGF is actually produced by mesenchymal cells. These are supporting connective tissue type cells found in the liver and also in other tissues. It's not produced by the hepatocytes that are actually doing the dividing.
A signal from supporting cells, then. What happens to HGF levels in the blood after, say, a partial hepatectomy?
According to Guyton and Hall, blood levels of HGF skyrocket. They can rise more than 20-fold after such a procedure.
If HGF is circulating everywhere in the blood, why does it mainly trigger regeneration only in the remaining liver tissue?
That's part of the mystery Guyton and Hall points out. The mitogenic response, the signal to divide, seems largely confined to the liver itself after these operations. It suggests that maybe HGF only gets activated locally in the injured organ, or perhaps the liver cells only become responsive to it under these specific conditions. The exact mechanism for this localized effect isn't fully clear.
So HGF is a major suspect for the go signal, but probably not the whole story.
Correct. Guyton and Hall mentions that other growth factors like epidermal growth factor, EGF, and certain cytokine signaling molecules like tumor necrosis factor, TNF, and interleukin-6, IL-6, may also be involved in stimulating the regenerative process. It's likely a complex interplay of signals.
Okay, so a bunch of factors likely tell the liver to grow. How does it know when to stop? What puts the brakes on once it reaches the right size?
Again, the source states that the factors involved in terminating the regeneration are also not well understood, but they highlight one factor in particular: transforming growth factor, or TGF-beta.
TGF-beta, what does it do?
TGF-beta is a cytokine, but unlike HGF, it's actually secreted by the hepatic cells themselves, and its effect is the opposite of HGF. It's a potent inhibitor of liver cell proliferation. Guyton and Hall suggest it might be the main terminator signal for liver regeneration.
So the liver cells might release their own stop signal once the job is done, a self-regulating system. Guyton and Hall also mentioned something intriguing about liver size being linked to overall body size.
Yes, experimental studies suggest that liver growth is somehow closely regulated by an unknown signal related to the organism's total body size.
Why would that be? What's the goal?
The apparent goal is to maintain an optimal liver-to-body weight ratio. This specific ratio seems crucial for ensuring the liver has the right capacity to handle the metabolic demands of the entire body it supports. It needs to be appropriately sized for the job.
So it doesn't just grow back to its original size. It seems to aim for a size that's proportional to the current body it's in. Fascinating. And finally, let's just reiterate that critical limitation on this amazing regenerative power.
Right. Guyton and Hall is clear. Conditions like fibrosis, scarring, ongoing inflammation, or persistent viral infections severely impair this regenerative process.
And if regeneration is impaired?
Then liver function progressively deteriorates. Despite its incredible potential, chronic insults can exhaust or disable this repair mechanism, leading inevitably to worsening liver disease.
So this phenomenal healing ability, driven by complex signals we're still figuring out, precisely controlled to start and stop, and even scaled to your body size, it's one of the liver's most impressive features. But sadly, it's a capacity that can be overwhelmed by persistent damage. Okay, with structure, circulation, and regeneration covered, let's move to section 4, the hepatic macrophage system. This focuses on the liver's role as a critical blood filter.
Yes, this brings us back to those Kupffer cells we talked about, lining the sinusoids. The liver plays a absolutely vital and perhaps sometimes overlooked role in cleaning the blood, especially the blood arriving directly from your intestines via the portal vein.
Why is filtering that specific blood so important?
Well, Guyton and Hall explains that as blood flows through the capillaries in your intestinal walls, it inevitably picks up large numbers of bacteria that normally live in your gut lumen. This is just a constant process.
And we have proof the liver intercepts these bacteria.
Oh yes, very compelling proof. Guyton and Hall states that if you take a blood sample from the portal vein before it enters the liver and culture it, you'll almost always grow colon bacilli common gut bacteria.
Okay.
But if you culture blood taken from the general systemic circulation blood that has already passed through the liver, you rarely find these bacteria. The difference is stark and points directly to the liver's filtration function.
That's a powerful comparison. The liver is acting like a gatekeeper, stopping gut bacteria from getting into the rest of the body. And it's the Kupffer cells doing the actual filtering.
Exactly. Guyton and Hall identifies those large phagocytic Kupffer cells as the key players. Lining the sinusoids, they're perfectly positioned to screen all the incoming portal blood.
Just how efficient are they at grabbing these bacteria? The source mentions some specific studies.
Yes, they mention studies using high-speed motion pictures that have visualized just how rapidly and efficiently these Kupffer cells cleanse the blood as it flows past them in the sinusoids.
What does that capture process look like at that speed? It must be quick.
It's incredible. Guyton and Hall describes it. When a bacterium makes even fleeting momentary contact with the surface of a Kupffer cell contact lasting less than 1/100th of a second, 0.01s, it's immediately pulled inward through the cell wall and becomes permanently trapped inside until the Kupffer cell digests it.
Less than 0.01 seconds of contact is enough for capture. That's unbelievably fast for a biological process. Do they manage to catch most of the bacteria coming through?
Guyton Hall gives an estimate of their success rate. It's thought that probably less than 1% of the bacteria entering the portal blood from the intestines actually succeed in passing all the way through the liver and into the general systemic circulation.
Less than 1% get through. That is a staggering level of filtration efficiency. It really underscores the liver's critical role in innate immunity, preventing constant systemic seeding of bacteria from our gut microbiome. It's like the ultimate high-speed security checkpoint.
It absolutely is. This blood cleansing function is crucial for preventing widespread infections originating from the gut.
So the liver is a master filter, an immune guardian. Now let's dive into what many consider its most complex and central role, its vast metabolic functions. Guyton Hall calls it a massive chemical factory.
That's a perfect description. The source refers to it as a large chemically reactant pool of cells with an exceptionally high rate of metabolism.
What does having such a high metabolic rate enable the liver to do?
It means the liver cells are constantly buzzing with complex biochemical reactions. They're sharing substrates and energy between countless different pathways. They're processing and synthesizing a huge variety of substances that are then shipped out to other parts of your body, and performing myriad other tasks essential for keeping your internal environment stable homeostasis.
Guyton Hall even comments on the sheer breadth of this.
Yes, they point out that a major chunk of the entire field of biochemistry is essentially devoted to studying the metabolic reactions that occur primarily in the liver. It's that central to body chemistry.
Okay, for this deep dive, we want to zero in on the major metabolic functions highlighted in the source, the ones crucial for understanding how the whole body works together physiologically. Let's start with carbohydrate metabolism. Guyton Hall lists 4 key functions here.
Right. The 4 main carbohydrate roles mentioned are 1. Storing large amounts of glycogen, 2. Converting other sugars like galactose and fructose into glucose, 3. Performing gluconeogenesis, making new glucose, and 4. Forming many other chemical compounds using intermediates from carbohydrate breakdown.
Let's focus on that first one, glycogen storage, and what the source calls the glucose buffer function. How does the liver act as a buffer for glucose?
This is incredibly important for keeping your blood glucose levels stable. So after you eat a meal containing carbohydrates, glucose gets absorbed from your gut, and the glucose level in the portal blood flowing to the liver rises. The liver then quickly removes much of this excess glucose from the blood.
And what does it do with it?
It converts it into glycogen, which is a storage form of glucose, like starch in plants. It essentially mops up the extra glucose and stores it.
Okay, so it stores it after a meal. What about between meals?
Then, when your blood glucose level starts to drop, say between meals or overnight, the liver breaks down that stored glycogen back into glucose and releases it into the bloodstream. Guyton and Hall calls this the glucose buffer function because it resists big changes. It prevents blood sugar from spiking too high after eating and from dropping too low when you're fasting.
What happens if this vital buffering system is broken, like in someone with significant liver damage?
Guyton Hall gives a clear example. In someone with poor liver function, after they eat a carb-rich meal, their blood glucose might shoot up 2 to 3 times higher than it would in someone with a healthy liver. The damaged liver just can't efficiently take up and store that incoming glucose load.
That really highlights the liver's central role in blood sugar control. Okay, what about the third function you listed, gluconeogenesis? What does that mean?
Gluconeogenesis is another critical way the liver maintains blood glucose. It literally means making new glucose. It's the liver's ability to synthesize glucose from non-carbohydrate sources.
When does this process become important? Is it always happening?
According to Guyton and Hall, it becomes significant primarily when your blood glucose concentration falls below normal levels, like during prolonged fasting or starvation. It's a key mechanism to prevent dangerously low blood sugar, hypoglycemia.
And what raw materials does the liver use to build this new glucose? It can't just make it out of thin air.
No, it uses other molecules. Primarily, it converts large amounts of amino acids, which come from the breakdown of proteins, and also glycerol, which is a component of fats, triglycerides, into glucose.
So it can turn protein and fat components into sugar.
Yes. This conversion process allows the liver to maintain a relatively normal blood glucose level even when you haven't eaten carbs for a while, ensuring a steady supply of glucose, especially for tissues like the brain, which heavily rely on it.
Amazing. It stores glucose, releases glucose, and makes new glucose from other stuff. It's the undisputed master regulator of blood sugar.
Okay, let's switch gears to fat metabolism. Guyton and Hall lists 3 specific important functions here too. Yes. For fat metabolism, the liver is key for 1. Oxidizing fatty acids to supply energy not just for itself, but for other body functions. 2. Synthesizing large quantities of cholesterol, phospholipids, and most lipoproteins. And 3. Synthesizing fat from excess proteins and carbohydrates.
Let's unpack that first one. Oxidizing fatty acids to supply energy for other body functions. How does the liver help fuel other tissues using fat?
Okay, so Guyton Hall explains the process. First, fats, triglycerides, are broken down into glycerol and fatty acids. These fatty acids are then taken up by the liver and rapidly broken down through a process called beta oxidation.
Beta oxidation, what does that produce?
It chops the long fatty acid chains into two carbon units, which form acetyl coenzyme A, or acetyl-CoA. Now, acetyl-CoA is a major fuel molecule that can enter the citric acid cycle to generate lots of energy.
Right. Does this happen only in the liver?
No, beta oxidation happens in many cells, but Guyton and Hall stresses that it occurs especially rapidly in liver cells. But here's the crucial twist. The liver processes fatty acids so fast that it generates more acetyl-CoA than it can actually use in its own citric acid cycle for its own energy needs.
So it makes more fuel than it can burn itself. What does it do with the excess acetyl-CoA?
Instead of burning it all, the liver converts much of this excess acetyl-CoA into something else. Two molecules of acetyl-CoA are condensed together to form acetoacetic acid.
Acetoacetic acid, what happens to that?
Acetoacetic acid is highly soluble in water. It readily diffuses out of the liver cells into the bloodstream and is transported throughout the body. Acetoacetic acid is one of the main ketone bodies.
And what do other tissues do with this acetoacetic acid arriving in the blood?
Other tissues, like muscle and brain, especially during fasting, readily absorb the acetoacetic acid from the blood. They then have the enzymes to convert it back into acetyl-CoA, which they can then feed into their own citric acid cycles to generate large amounts of energy.
Ah, I see. So the liver acts like a central processing hub. It rapidly breaks down fatty acids into this transportable fuel source ketone bodies like acetoacetic acid, which it then exports for the rest of the body to use as energy.
Exactly. It shows the liver is responsible for a major part of processing fat energy for the entire body, not just for itself. Okay, let's move to the second fat function: synthesis of cholesterol and phospholipids.
Right, building essential fats.
The liver is the primary site for synthesizing these crucial lipids. Guyton and Hall notes that about 80% of the cholesterol made in the liver gets converted into bile salts, essential for digestion, and secreted into the bile.
And the other 20%.
The remaining cholesterol, along with phospholipids synthesized in the liver, gets packaged into particles called lipoproteins. These lipoproteins are then transported via the blood to all the other tissues and cells in the body.
What do the body's cells use this liver-made cholesterol and phospholipids for?
They're absolutely essential building blocks. Guyton and Hall explains they're used by cells everywhere to construct cell membranes, membranes around organelles inside the cells, and they also serve as precursors for making other vital substances, like steroid hormones.
So the liver is manufacturing and distributing the basic lipid components needed by every cell. What about the third function, synthesizing fat from other types of nutrients?
Yes, Guyton and Hall states that almost all the synthesis of fat, triglycerides in the body, that occurs when you consume excess carbohydrates or proteins happens in the liver.
So if you eat too many carbs or too much protein, the liver is the main place where that excess energy gets converted into fat for storage.
Precisely. After the liver synthesizes this fat, it packages it into lipoproteins, specifically VLDL, very low-density lipoproteins, and transports it through the bloodstream to your adipose tissue, your fat cells where it's stored.
Incredible. It's taking all incoming nutrients, deciding whether to burn them immediately, store them short-term as glycogen, or convert them long-term into fat, and then shipping that fat off for storage. Truly the metabolic control center.
Okay, let's move to protein metabolism. Guyton and Hall makes a very stark statement about the liver's importance here.
They really do. They state very clearly that the body cannot dispense with the liver's contribution to protein metabolism for more than just a few days without death ensuing. That really hammers home how absolutely indispensable its role is here.
A few days without it, and that's it. Wow. What are the four most critical protein functions listed?
According to the source, the four most important are: 1. Deamination of amino acids. 2. Formation of urea to get rid of ammonia. 3. Formation of plasma proteins. And 4. Interconversions between different amino acids and synthesis of other compounds from amino acids.
Okay, let's start with deamination. What is that and why is it necessary? Deamination is basically removing the amino group, the nitrogen-containing part, from an amino acid molecule. Guyton and Hall explains this step is required before the remaining carbon skeleton of the amino acid can be used by the body for energy, or before it can be converted into glucose or fat.
Does this happen anywhere else?
A small amount can occur elsewhere, like in the kidneys, but Guyton and Hall emphasizes this is much less important than the massive amount of deamination carried out by the liver. The liver is overwhelmingly the main site for this.
Got it. Removing the nitrogen part. And that nitrogen has to go somewhere, which leads directly to the second function, urea formation. What happens to that toxic ammonia that's generated?
This is arguably one of the liver's most critical detoxification functions. It forms urea, which is essential for removing highly toxic ammonia from your body fluids.
Where does all this dangerous ammonia come from?
Well, large amounts are produced during that deamination process we just talked about, but also significant amounts are continually produced by bacteria living in your gut, and this ammonia gets absorbed into the portal blood flowing to the liver.
And why is getting rid of it so crucial? What happens if the liver fails to make urea?
Guyton and Hall states the severe consequence. If the liver doesn't form urea, ammonia levels in the plasma rise rapidly. Excess ammonia is extremely toxic, especially to the brain, and quickly leads to a state called hepatic coma and eventually death. Converting ammonia to the relatively harmless urea, which can then be safely excreted by the kidneys and urine, is absolutely life-sustaining.
The liver is literally saving us from constant ammonia poisoning. The source also mentions that even just poor blood flow to the liver can cause problems here.
Yes, even if the liver cells are okay, if blood flow through the liver is greatly decreased, for example, if someone develops a shunt that bypasses the liver, connecting the portal vein directly to the vena cava, the liver doesn't get a chance to remove the ammonia effectively. Ammonia levels can build up and cause severe toxicity. It highlights how important it is for essentially all that portal blood carrying gut-derived ammonia to pass through the liver's processing system.
Right, bypassing the factory means the toxins don't get neutralized. Okay, function number 3, formation of plasma proteins.
This is another area where the liver is dominant. Guyton and Hall states that essentially all the plasma proteins circulating in your blood, with the notable exception of a part of the gamma globulins, which are antibodies, are manufactured by the hepatic cells.
Essentially all. What percentage are we talking about?
It accounts for about 90% of all plasma proteins. Think about proteins like albumin, which maintains fluid balance, fibrinogen and prothrombin, essential for blood clotting, and many others. They're almost exclusively made by the liver.
And the gamma globulins, the antibodies, where are they made?
Guyton and Hall clarifies they're mainly formed by specialized immune cells called plasma cells, located in lymph tissues throughout the body. But the vast bulk of other circulating proteins originates from the liver.
How quickly can the liver churn out these essential proteins?
Guyton Hall gives a maximum rate of about 15 to 50 grams per day. That's a substantial amount of protein synthesis.
It is. What happens if plasma protein levels drop too low, maybe due to illness or protein loss? Does the liver respond?
Yes, it does. Guyton and Hall notes that low plasma protein levels act as a powerful stimulus for the liver. It actually triggers rapid mitosis cell division of the hepatocytes, causing the liver to grow larger. At the same time, the liver rapidly increases its output of plasma proteins until the concentration in the blood returns to normal.
So low protein tells the liver to grow bigger and make more protein, another example of its dynamic response. What's the clinical flip side? What happens in chronic liver disease like cirrhosis?
Guyton Hall points out that in chronic liver disease, the liver's ability to synthesize these proteins is severely impaired. As a result, plasma protein levels, especially albumin, can fall very low.
And the consequence of low albumin.
Since albumin is crucial for maintaining osmotic pressure to hold fluid inside blood vessels, low albumin leads to fluid leaking out into the tissues, causing generalized edema, swelling, and specifically contributes significantly to ascites, that fluid buildup in the abdomen we talked about earlier. It's a direct link.
So, the ascites in liver disease is partly due to high pressure forcing fluid out, and partly due to low protein synthesis failing to keep fluid in. It connects back perfectly. Okay, the fourth protein function, interconversions of amino acids and synthesis of other compounds.
Right. This is about the liver's ability to synthesize certain amino acids itself, and also to create other important chemical compounds using amino acids as building blocks.
Which amino acids can the liver make?
It synthesizes the so-called non-essential amino acids. These are the ones your body can produce, meaning you don't absolutely have to get them from your diet, though you usually do. The liver is the primary site for manufacturing these.
How does it actually build a new amino acid? What's the process?
Guyton and Hall gives a glimpse of the biochemistry. To make a non-essential amino acid, the liver first synthesizes a molecule called a keto acid, which has the right carbon backbone structure but lacks the crucial amino group. Then, a key reaction called transamination occurs.
Transamination, okay.
An amino group is transferred from an existing available amino acid, often one from your diet, onto that keto acid. This replaces a keto oxygen on the keto acid with the amino group, thereby forming the new complete amino acid.
So it's like shuffling the amino groups around between different carbon skeletons to create the specific amino acids the body needs at that moment.
Exactly. It's a fundamental way the body manages its amino acid pool, ensuring the right building blocks are available, especially for those amino acids it can manufacture itself.
Okay, so protein metabolism in the liver is incredibly diverse and critical.
Yeah.
Breaking down amino acids for energy, detoxifying the resulting ammonia into life-saving urea, synthesizing almost all essential plasma proteins, and interconverting amino acids. Absolutely indispensable. Let's move to section 6, other vital functions, focusing on storage and excretion. The liver is famous as a storage depot, too.
It certainly is. Guyton and Hall highlights the liver's particular propensity for storing vitamins. This is why, historically, eating liver was known as an excellent way to treat certain vitamin deficiencies.
Which vitamins does it store particularly well, and for how long can these reserves last?
Guyton Hall gives some impressive examples of its capacity. Vitamin A, stored in the greatest quantity. The liver can hold enough vitamin A to prevent deficiency for up to 10 months. Vitamin D, large amounts are also stored, typically enough for 3 to 4 months. Vitamin B12. It stores enough B12 to last for at least 1 year and possibly even several years.
Months, even years of supply for key vitamins buffered in the water. Amazing long-term storage. What about iron?
The liver is also the main storage site for iron in the body. Guyton Hall states that, apart from the iron actually incorporated into hemoglobin inside red blood cells, by far the largest proportion of your body's iron is stored in the liver.
And how is it stored? What's the mechanism?
It's stored primarily in a protein complex called ferritin. Liver cells contain large amounts of a protein called apoferritin. Guyton Hall explains that apoferritin can bind reversibly with iron.
Reversibly meaning you can grab iron and let it go.
Exactly. That reversibility is key. It allows this apoferritin-ferritin system to function as a dynamic storage pool. When you have excess iron circulating in your blood, it readily combines with apoferrin inside liver cells to form ferritin, effectively storing the iron safely.
And when iron levels are low,
When iron levels in the circulation drop and your body needs more iron, for example, to make new red blood cells, the ferritin releases the stored iron back into the circulation.
so the liver's ferritin system is actively managing and buffering blood iron levels, preventing both toxic overload and deficiency.
Precisely. Guyton and Hall summarizes it as acting like a crucial blood iron buffer, as well as a long-term storage depot. It's constantly adjusting iron levels.
Another buffer, glucose, blood volume, iron. What about blood coagulation factors? We mentioned protein synthesis, but this is critical.
Yes, this is another vital synthetic function tied to protein metabolism. The liver manufactures several substances that are absolutely essential for your blood to clot properly. Guyton and Hall lists key ones like fibrinogen, prothrombin, accelerator globulin, factor V, factor 7, and several other important factors.
And isn't there a specific vitamin the liver needs to make some of these clotting factors?
Yes, absolutely. Vitamin K is required by the liver's metabolic machinery to synthesize several of these crucial factors specifically mentioned are prothrombin and factor 7, IX, and X.
What happens if someone is deficient in vitamin K, or if their liver is too damaged to use it properly?
The consequences are serious for blood clotting. Guyton Hall states that without sufficient vitamin K activity in the liver, the concentrations of these vitamin K-dependent clotting factors in the blood decrease markedly. This can impair blood coagulation so severely that it almost prevents it altogether, leading to potentially life-threatening bleeding problems.
So liver health and adequate vitamin K are directly linked to preventing excessive bleeding, a critical connection. Lastly, under this section, the liver is well known for removing or excreting various substances.
Yes, Guyton Hall highlights its extensive ability to detoxify or excrete many drugs and hormones, often by secreting them into the bile or by chemically modifying them so they can be excreted by the kidneys.
Can you give some examples of drugs the liver excretes into bile?
Guyton and Hall lists several common antibiotics, including sulfonamides, penicillin, ampicillin, and erythromycin, as examples of drugs that are significantly excreted by the liver into the bile. They then pass into the intestines and are eliminated in the feces.
What about hormones? Does the liver clear hormones from the body?
Yes, it plays a major role in inactivating or clearing several important hormones. Mentioned are thyroid hormones like thyroxine and essentially all the major steroid hormones such as estrogen, cortisol, and aldosterone. The liver chemically alters or excretes them.
What's the clinical significance if the liver is damaged and can't clear these hormones effectively?
Guyton and Hall notes this can lead to the excessive accumulation of one or more of these hormones in the body fluids. This buildup, in turn, can cause overactivity of the corresponding hormonal systems, leading to a variety of symptoms depending on which hormone is accumulating. It's a crucial point. Poor liver function can manifest as hormonal imbalances.
That's a key link. Unexpected hormone issues could potentially point back to liver problems. And one final excretion mentioned is for calcium.
Yes, Guyton Hall states that one of the major routes for eliminating excess calcium from the body is through secretion by the liver into the bile. The calcium then enters the gut and is lost in the feces.
So the liver even has a hand in mineral balance.
Wow.
Storing months or years of vitamins, actively buffering iron, making essential clotting factors, clearing drugs and hormones, even helping excrete calcium. The liver is undeniably a central processing hub, managing inventory and waste disposal like nothing else.
It's constantly working behind the scenes to keep your internal chemical environment within very narrow, safe limits.
That leads us perfectly into our final section, Section 7, bilirubin, bile, and jaundice. This is such a classic example of how understanding liver physiology gives us a direct, often visible diagnostic window.
Absolutely. The story of bilirubin is, as Guyton Hall puts it, an exceedingly valuable tool for diagnosing various diseases, especially those affecting red blood cell breakdown, hemolysis, and different types of liver diseases.
Before we dive into bilirubin itself, just briefly, what's the general role of bile that the liver produces?
Well, Guyton and Hall mentions its main role in digestion. The bile salts within bile are crucial for breaking down and absorbing fats in the intestine, which they cover elsewhere. But another key function of bile is simply as an excretion route. The liver dumps various waste products and substances into the bile to be eliminated from the body via the feces.
And bilirubin is a major one of these substances excreted in bile. What exactly is bilirubin?
Bilirubin is described as a greenish-yellow pigment. Guyton Hall identifies it as a major end product generated from the breakdown of hemoglobin, the molecule inside red blood cells that carries oxygen.
So it's basically a waste product from the normal turnover of old red blood cells. Let's trace the path of bilirubin from its formation to its elimination, as Guyton and Hall lays it out. Where does the process begin?
It starts with the red blood cells themselves. They have a limited lifespan, typically around 120 days. As they get old, their outer membranes become fragile and eventually rupture.
Releasing the hemoglobin inside. What happens to that released hemoglobin?
It gets quickly engulfed or phagocytized by macrophages, specialized immune cells found throughout the body's tissues. This network of macrophages is called the reticuloendothelial system, and it includes those Kupffer cells we already discussed lining the liver sinusoids.
Okay, so macrophages eat the old hemoglobin. What breakdown happens inside the macrophage?
Inside the macrophage, the hemoglobin molecule is split into its two main parts. Globin, which is the protein part and gets broken down into amino acids for recycling, and heme, which is the iron-containing pigment part. The ring structure of the heme molecule is then opened up.
What does opening the heme ring produce?
It yields two main things. First, free iron, which is released and gets transported in the blood bound to a protein called transferrin, ready for reuse. Second, what's left is a straight chain of four smaller rings called chiral nuclei. This straight chain is the basic structure that will be converted into bilirubin.
So globin protein and iron get salvaged, and the core heme structure starts the bilirubin pathway. What's the first pigmented substance formed from that chiral chain?
The first substance formed is called biliverdin, which has a greenish color. Guyton and Hall states that biliverdin is then rapidly reduced, chemically changed.
Reduced to form what?
To form free bilirubin, which is also often called unconjugated bilirubin. This unconjugated bilirubin is then gradually released from the macrophages into the bloodstream, plasma.
How does this free or unconjugated bilirubin travel in the blood? Is it soluble enough to dissolve in plasma?
No, it's not very water-soluble on its own. Guyton and Hall explains that as soon as it enters the plasma, unconjugated bilirubin binds very tightly to plasma albumin, a major protein in the blood. It's transported throughout the blood and body fluids attached to albumin.
So albumin acts like a taxi, carrying the unconjugated bilirubin around. Where does it take it?
It takes it to the liver. Within a few hours of being released, the albumin-bound unconjugated bilirubin reaches the liver sinusoids and is absorbed through the membrane of the hepatic cells, hepatocytes. Once inside the liver cell, it gets released from its albumin carrier.
And once inside the liver cell, the liver modifies it.
Yes, this is the absolutely critical step called conjugation. Soon after entering the hepatocyte, the unconjugated bilirubin gets conjugated, meaning other molecules are attached to it to make it more water-soluble.
What molecules does the liver attach?
Guyton Hall gives the typical breakdown. About 80% is conjugated with glucuronic acid, forming bilirubin glucuronide. About 10% is conjugated with sulfate, forming bilirubin sulfate, and the remaining 10% with various other substances.
So the liver basically sticks water-soluble tags onto the bilirubin. What's the purpose of making it water-soluble?
It allows the liver to actively secrete it into the bile. In these conjugated forms, bilirubin is pumped out of the hepatocytes by an active transport process directly into those tiny bile canaliculi we talked about in the liver's structure.
Okay.
From the canaliculi, it flows into the larger bile ducts, and eventually, the bile containing this conjugated bilirubin travels down into the intestines.
Right, let's recap that path. Old red cells break down. Macrophages release unconjugated bilirubin. Travels bound to albumin. Liver cells take it up. Liver conjugates it, makes it water-soluble. Liver actively secretes conjugated bilirubin into bile. Bile flows into the gut. What happens to the conjugated bilirubin once it reaches the intestines?
In the intestines, bacteria that normally live there go to work on it. About half of the conjugated bilirubin gets converted by bacterial action into a substance called urobilinogen. Guyton and Hall notes that urobilinogen is highly soluble.
Does all this urobilinogen just pass out in the feces?
No, not all of it. Because it's soluble, a significant portion of the urobilinogen gets reabsorbed from the intestine back through the intestinal lining into the portal blood.
Ah, so some of it loops back into the bloodstream. What happens to the urobilinogen that gets reabsorbed?
Most of that reabsorbed urobilinogen goes straight back to the liver via the portal vein, and the liver promptly takes it up and re-excretes it back into the bile. It's called the enterohepatic circulation of urobilinogen.
So if most goes back to the liver, what about the rest?
A small fraction, about 5% of the reabsorbed urobilinogen, manages to bypass the liver's uptake and continues circulating in the blood until it reaches the kidneys. The kidneys then filter this urobilinogen out and excrete it into the urine.
So a tiny amount of urobilinogen normally ends up in the urine. What about the urobilinogen that doesn't get reabsorbed from the gut, or the stuff that gets re-excreted by the liver? What gives urine and feces their characteristic colors?
Guyton and Hall explains that urobilinogen itself is colorless, but it gets converted into colored pigments. In the urine, after it's exposed to air, urobilinogen gets oxidized to form urobilin, which contributes to urine's yellow color.
Okay.
And in the feces, the urobilinogen that remains in the gut gets chemically altered and oxidized by bacteria to form stercobilin, which is the pigment primarily responsible for the characteristic brown color of normal feces.
Urobilinogen, urobilinin, and feces, those are the final colored breakdown products originating from hemoglobin.
Exactly. The whole pathway, from hemoglobin in old red cells to the final colored pigments in waste products, is clearly traced by Guyton and Hall. It shows how bilirubin is normally processed and eliminated.
Now, what happens when something disrupts this pathway, causing bilirubin to accumulate in the body? This leads to jaundice, right?
Yes, jaundice is the clinical term for the yellowish discoloration that appears in body tissues. It's usually most noticeable in the skin and the sclera, the whites of the eyes, but deeper tissues can be affected too.
And what causes that yellow color?
It's caused by the buildup of large quantities of bilirubin in the extracellular fluids throughout the body. Depending on the cause of the problem, this buildup could be mostly unconjugated bilirubin, mostly conjugated bilirubin, or a mixture.
What's the normal level of bilirubin in the blood?
Guyton and Hall states that the average normal plasma concentration is quite low, around 0.5 milligrams per deciliter, mgdl. And at this normal level, nearly all of it is the unconjugated form, bound to albumin.
How high can bilirubin levels get when things go wrong?
In certain abnormal conditions, the plasma bilirubin can soar, sometimes reaching as high as 40 mg/dl. And in these very high levels, a significant proportion is often the conjugated type.
At what level does the yellowing actually become visible as jaundice?
Guyton and Hall notes that the skin typically starts to look noticeably yellow or jaundiced when the total plasma bilirubin rises to about 3 times the normal level, so somewhere above 1.5 mg/dl.
So even a modest increase above normal can cause visible signs. What are the two main categories of causes for jaundice that Guyton Hall outlines?
They list 2 principal mechanisms. First, increased destruction of red blood cells. hemolysis, which releases bilirubin into the blood faster than the liver can possibly excrete it. And second, obstruction of the bile ducts or significant damage to the liver cells themselves, which prevents even the normal amounts of bilirubin from being properly excreted from the liver into the gut.
And these two types have specific names.
Yes, the first type is called hemolytic jaundice, and the second type is broadly termed obstructive jaundice, which includes jaundice caused by liver cell damage, sometimes called hepatocellular jaundice.
Okay, let's break down hemolytic jaundice first. What's the core problem causing the bilirubin backup here?
In hemolytic jaundice, the fundamental issue is that red blood cells are breaking down much too rapidly. This floods the system with unconjugated bilirubin being released from macrophages, overwhelming the liver's capacity to take it up, conjugate it, and secrete it into the bile.
So the liver's processing machinery might be working perfectly normally. It's just getting swamped by the sheer volume of incoming unconjugated bilirubin from all the broken down red cells.
Exactly. Guyton Hall makes the point that the liver's excretory function itself is not necessarily impaired in pure hemolytic jaundice. It's simply the rate of bilirubin formation that is dramatically increased.
Given that, what type of bilirubin predominantly builds up in the blood in hemolytic jaundice?
Because the liver is being presented with an excessive load of unconjugated bilirubin and can't keep up with processing it, the plasma concentration of free unconjugated bilirubin rises significantly above normal. That's the biochemical hallmark.
And what happens downstream in the gut and urine with urobilinogen in this situation?
Since much more bilirubin, even if processed slower, eventually reaches the intestine, the bacteria there produce a greatly increased amount of urobilinogen. Consequently, more urobilinogen is reabsorbed back into the blood than usual, and therefore a larger quantity ends up being excreted by the kidneys into the urine. High urine urobilinogen is another sign.
Okay, so hemolytic jaundice recap. Too many red cells breaking down liver gets overwhelmed. Unconjugated bilirubin piles up in the blood. Lots of urobilinogen is made in the gut. High levels of urobilinogen appear in the urine. Now, let's contrast that with obstructive jaundice. What causes this type?
Obstructive jaundice results either from a physical blockage of the bile ducts, maybe a gallstone lodged in the common bile duct, or a tumor pressing on it, or from significant damage to the liver cells themselves, like what happens in viral hepatitis or cirrhosis, which impairs their ability to secrete bile.
Is the rate of red blood cell breakdown and initial bilirubin formation normal in obstructive jaundice?
Yes, typically. Guyton and Hall notes that bilirubin formation is usually normal. The problem isn't making too much bilirubin. The problem is that the bilirubin that is made and processed by the liver cannot get out into the intestines effectively, either because the exit path is blocked or the liver cells are too sick to pump it out.
So what happens to the unconjugated bilirubin arriving at the liver from the blood? Does it still get processed?
Guyton and Hall explains that, yes, the unconjugated bilirubin still enters the liver cells. And assuming the cells aren't completely destroyed, it usually gets conjugated normally into the water-soluble forms, bilirubin, glucuronide, etc. The conjugation machinery itself is often still functional, at least initially.
But if it gets conjugated normally but then can't be secreted out into the bile ducts because of the obstruction or cell damage, where does that conjugated bilirubin go?
It gets trapped. The conjugated bilirubin builds up inside the liver cells and the tiny bile canaliculi. Guyton and Hall suggests it then essentially leaks or is regurgitated back into the bloodstream. This might happen through the rupture of congested canaliculi, emptying directly into the liver's lymph vessels, which then drain into the general circulation.
So the conjugated bilirubin, unable to exit forwards into the gut, backs up and spills backwards into the blood.
That's a good way to think about it. And the critical diagnostic consequence is that in obstructive jaundice, most of the bilirubin that accumulates in the plasma is the conjugated type.
Ah. So this difference in the type of bilirubin that's elevated in the blood, primarily unconjugated in hemolytic jaundice versus primarily conjugated in obstructive jaundice, is the key distinction.
It's a fundamental difference. Guyton and Hall emphasizes that standard laboratory blood tests can easily differentiate between the unconjugated, often called indirect, and conjugated direct bilirubin fractions in plasma. This is crucial for diagnosis.
Guyton Hall even mentions a specific older test name associated with this.
Yes, the Vandenberg reaction is mentioned as a classic test used to make this differentiation.
What if the bile duct obstruction is complete? Are there other clues we can get from tracing the rest of the bilirubin pathway?
Yes, definitely. If there's a total blockage, then no bilirubin whatsoever can reach the intestines.
And if no bilirubin reaches the intestines?
Then the gut bacteria cannot form any urobilinogen.
And if no urobilinogen is formed in the gut.
Then none can be reabsorbed into the blood, and consequently, none can be excreted by the kidneys into the urine. So, Guyton and Hall states that in complete obstructive jaundice, tests for urobilinogen in the urine will be completely negative.
That's a very strong diagnostic sign. Zero urine urobilinogen points towards a total blockage. What about the appearance of the patient's stool in this case?
The stools typically become pale or clay-colored. This is because the normal bile pigments, especially stercobilin, which gives feces their brown color, are completely absent since no bile is reaching the gut.
Clay-colored stools plus absent urobilinogen is likely complete obstruction. Is there one more difference related to the kidneys and the type of bilirubin?
Yes, and it provides another useful diagnostic clue, sometimes even observable. Guyton Hall points out that the kidneys can filter and excrete small amounts of the water-soluble conjugated bilirubin if its level gets high enough in the blood. However, the kidneys cannot excrete the unconjugated bilirubin because it's tightly bound to albumin, which is too large to be filtered easily.
So if someone has high levels of conjugated bilirubin in their blood, as happens in obstructive jaundice.
Then significant amounts of this conjugated bilirubin can spill over into the urine, making the urine appear dark or tea-colored.
And the simple bedside test for this.
Guyton Hall mentions the classic simple test. If you shake a sample of the urine, and it contains a lot of conjugated bilirubin, the foam that forms on top will have an intense yellow color.
Right, the bilirubinuria test. So putting it all together. By understanding this whole bilirubin pathway, how it's formed, transported, conjugated, excreted into bile, processed in the gut, and cleared by the kidneys, and by using relatively simple observations and tests, like checking plasma bilirubin types, urine urobilinogen, stool color, urine color foam, clinicians can effectively differentiate between jaundice caused by too much red cell breakdown versus jaundice caused by problems within the liver or bile ducts, and even get clues about the severity, like whether an obstruction is complete.
Absolutely. Guyton and Hall uses a bilirubin as a prime example of how knowing the fundamental physiology provides incredibly valuable diagnostic power. Bilirubin acts like a biological tracer, revealing where the pathway is disrupted.
Wow. What a journey through the liver today. We've really gone deep, unpacking it layer by layer, drawing on all that detail from Guyton and Hall. We started with the intricate anatomy, the lobules, sinusoids, Kupffer cells, spaces of Dissy built for massive processing. We explored the vascular system, the huge blood flow, low resistance, portal hypertension aside its role as a blood reservoir. We marveled at its almost unbelievable regeneration, driven by HGF and TGF-beta, yet vulnerable. We dove into its absolute centrality in metabolism, mastering carbs, fats, and proteins. We covered its vital storage of vitamins and iron and its critical excretion of drugs, hormones, and even calcium. And finally, that fascinating story of bilirubin, showing how physiology directly informs diagnosis of jaundice.
It's truly clear, isn't it? Every detail, from the microscopic structure to the body-wide consequences of dysfunction, just hammers home the liver's status as arguably the body's most complex and vital chemical processing plant. And really digging into these mechanisms with the depth provided by texts like Guyton Hall is what helps you move towards that elite professional level of understanding we talked about.
Absolutely. Going this deep really reveals the stunning interconnectedness and the sheer weight of responsibility the liver carries. And, considering just how many indispensable metabolic jobs it performs, plus that unique ability to regenerate, yet knowing how susceptible both are to chronic inflammation or damage, it really leaves you with the question, doesn't it? Can we ever truly hope to replicate something this complex, this integrated artificially? Or are we ultimately perhaps forever reliant on the intricate, natural balance of the liver itself?
That is a profound question, and it's exactly what drives so much ongoing research in areas like bioartificial livers and tissue engineering. The liver's natural capabilities are truly a benchmark of biological design.